Hi, Chris Good here, and this is the second in our Core Concepts video series for Drugs, Behavior, and Society, PSYC2050. In the first video, we talked about the action potential, and in this video, we're going to talk about synaptic transmission and neurotransmission. That's communication between two neurons. In the first video, this is the last thing that we looked at, was this graph of the action potential over time. And remember that we started off with a single neuron at rest. It's sitting here right around minus 70 millivolts or so. And then something happened to increase the, or not increase the voltage, but to slightly depolarize the cell, to move the voltage of the cell closer to 0, to move it in the positive direction. And we didn't say what that was. So what was the thing that started the voltage to come up in a positive direction in the first place? Now, once it reached the threshold for those voltage-gated sodium channels, sodium comes rushing into the cell, but something had to get it up there to begin with. The rest of the action potential goes that the sodium comes rushing into the cell, that drives the voltage up to a positive voltage, actually crosses 0 and starts to polarize again in the opposite direction, at which point the sodium channels close, potassium channels open, potassium comes rushing out of the cell, taking its positive charge with it, and driving the voltage back down until it stabilizes around its resting potential. But what was it early on that started the whole action potential? Well, here's our neuron again, and remember that neurons have a cell body, they have a nucleus. Most neurons have some kind of dendritic branching up here attached to the cell body, and there's some kind of axon that comes out of most neurons, and it terminates in these branches that end in the axon terminals, the ends of the axon. That action potential is that signal that's sent within a single neuron from one end to the other, and the other end can sometimes be very far away. So that action potential has got to travel really fast. The action potential is generated here at the beginning of the axon in a region called the axon hillock, and there's lots and lots of voltage-gated sodium channels here in that region where the action potential is generated. So if the action potential is a signal sent within a neuron, then how does the signal get to another neuron? Because remember, neurons do two things. They receive signals from other neurons, and they send signals to other neurons. Well, let's invoke another neuron here. So up at the top, we have, now they're yellow neurons. We have a neuron that's going to be sending a signal up here that the action potential goes all the way down here to the axon terminals. And you'll see those terminals come very close to, but don't quite touch, a second neuron. This is the neuron that's going to be receiving the signal. And with all the branching of the axon at the end here, I see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 terminal boutons that come very close to the cell body, and the dendrites of this receiving neuron, the neuron that's going to receive the signal. If you look inside this red box, we have an exploded view down here in the center of the slide that shows you a single terminal bouton and how it comes very close to kind of an upswelling of the receiving neuron's cell membrane. And this whole arrangement of a terminal bouton contacting the second receiving neuron, we call this whole thing the synapse. The synapse. And you'll see, if you look in this black box here, it's an exploded view onto the right, there's a space in between the two cells. There's a space between the sending neuron and the receiving neuron. We call this the synaptic cleft or gap. It isn't just space, it's filled with fluid and some other stuff as well. We'll get to that in just a second. So if this whole thing is called the synapse, the space is called the synaptic cleft or gap, then we can talk about the sending cell as being presynaptic, right? It's on one side of the synapse. And the receiving cell as being postsynaptic. So the presynaptic neuron is going to send a signal to the postsynaptic neuron. And that signal comes in the form of a chemical message. So all these little bubbles that you see in here are actually membrane-bound organelles that are called vesicles. And if you look inside the exploded view again, you can see that there are molecules inside of these vesicles. They're called synaptic vesicles, and the molecules inside are called neurotransmitters. These are the chemicals that are going to carry the message from the presynaptic neuron to the postsynaptic neuron. How does it get there? Well, when this sending neuron generates an action potential, that positive going spike in voltage reaches all of the axon terminal boutons. It comes down into each one of them through the process that we've discussed in the previous video. And remember, that's a positive going voltage spike that propagates down because of voltage-gated sodium channels. Well, there are other channels here that are proteins expressed in the axon terminal that pass other types of ions, namely calcium. There are voltage-gated calcium channels in the membrane here, and when the positive going voltage spike activates them, they open their pore and calcium comes into the cell. Calcium interacts with proteins that are expressed here in the presynaptic membrane and here on the synaptic vesicles to dock the vesicle to the presynaptic membrane and fuse those membranes together so that basically this bag of chemicals empties out into the space. So the neurotransmitter molecules then come into the synaptic gap because calcium entered this cell through voltage-gated calcium channels and enabled proteins on the membranes of both the vesicles and the presynaptic membrane to basically fuse these two things together. So transmitter molecules get dumped out here. All right, that's the signal that is sent, but how is that signal received? In order to receive the signal, this postsynaptic neuron must have receptors, and these receptors are large proteins that are embedded in the membrane, and they have a binding site here, a binding site that's shaped just for a specific neurotransmitter molecule. So when those neurotransmitters come across, they diffuse and then they fit into their binding site. There's lots and lots of different types of receptor proteins, and they do many different things to the postsynaptic cell. This is just one example of a type of receptor protein that actually forms an ion channel. And in this case, it's a sodium channel. These are not like the voltage-gated sodium channels that we talked about in the action potential, but they are gated. They're not gated by voltage, they're gated by the presence of a neurotransmitter molecule. So we could say they're transmitter-gated receptor channels for sodium. We could say they're ligand-gated receptors because a ligand is just something that fits into a binding site. And in this case, the ligand is a neurotransmitter. You already know the names of a lot of different neurotransmitters like dopamine and serotonin and how about acetylcholine? Let's say that this neurotransmitter molecule is acetylcholine, and this is a receptor that is a binding site for acetylcholine. So this is an acetylcholine receptor, and when acetylcholine binds, that opens this particular channel. That's actually how one type of acetylcholine receptor works. So acetylcholine sticks here, the pore opens up, sodium comes into the cell for the same reason that it wanted to be in the cell for the action potential. There's less sodium in the cell than there is outside of the cell. And remember, if this cell is at rest, there's a negative voltage, so positively charged sodium is going to be attracted down its a concentration gradient, as well as a charge gradient if there's negative charge on the inside. All right, sodium is permitted to go in the cell only because acetylcholine is bound to the receptor channel. So sodium enters the cell. Well, what does that do to the voltage of this cell? It makes it go more positive. And if enough acetylcholine is released at enough of these different synapses, then enough sodium may enter the cell to bring the postsynaptic cell to threshold for its voltage-gated sodium channels here in the axon hillock, and it may fire an action potential. So remember, we wondered where that increase in voltage, right, where that depolarizing voltage came from in the first place in this sending neuron that fired an action potential. Well, it was probably some other neuron up here that released transmitter onto the dendrite stem cell body enough so that enough sodium or some other positively charged ion entered the cell to raise the voltage to threshold for its voltage-gated sodium channels to let sodium into the cell. There are other types of transmitters with receptors expressed in the postsynaptic neuron that have different actions, and not all of them pass an ion into the cell. But let's talk about a case where there is one. There's a neurotransmitter called GABA, that stands for gamma-aminobutyric acid, and when it's released and it interacts with a GABA receptor, right, just in a very similar way, it isn't sodium that enters the cell. There are receptor ion channels, right, for that bind GABA to them, but it's a negatively charged ion that enters the cell. In fact, it's a chloride anion that carries a single negative charge. What would that do to the voltage here in the postsynaptic neuron? Well, if enough chloride comes in, then that voltage is going to go even more negative than it is when it's at rest. So maybe from minus 70 millivolts down to minus 75. It's going to drive it further away from the threshold for its voltage-gated sodium channels and make it less likely to fire an action potential. All right. So in the next slide is just another depiction of what happens here. The action potential would come down the axon. Voltage-gated calcium channels would allow calcium to enter the cell, and that calcium would interact with proteins on the vesicles and in the presynaptic membrane to spill the contents out into the synaptic gap between an axon terminal bouton and the postsynaptic neuron that has receptor proteins, and they have little binding sites in there for their specific receptor. When it sticks here, the pore will open, and any sodium that is in the gap will enter the cell if that's the ion that's passed by this receptor. So whether the ion is positive or negative depends on the receptor that's expressed, and sometimes on the transmitter that is released. There's some transmitters like acetylcholine that usually interact with receptor proteins that pass positively charged ions. Some like GABA that interact with receptor proteins that usually pass chloride, negatively charged ions, and so on and so forth. Why do we mention this in a drugs course? Because these are the places where drugs can act to change what neurons are doing, to change the way that they're firing, and ultimately change our behavior. So there's two places where drugs can act. They can act on the presynaptic neuron that's sending the signal. They can act on the postsynaptic neuron that's receiving the signal. And let's talk about some different mechanisms of action, some different routes of drug action that can be taken at both of these. We'll start with the presynaptic. And there's lots of different places, so bear with me. First of all, these transmitter molecules that are released from the presynaptic neuron must be synthesized in the cell, packaged into vesicles and transported down the axon into the terminal bouton. So drugs can affect the synthesis of neurotransmitters. They could block the synthesis. They can increase the synthesis. There are motor proteins that grab onto these vesicles and pass them down the axon. So axonal transport can be interfered with by drugs, sped up or slowed down. There are drugs that can block the conduction of action potentials or enhance it. For example, this is a voltage-gated sodium channel that you see here in blue, and sodium can't enter the channel because it's being blocked by a drug compound. There's a couple of different drugs that work this way. One is made by the blowfish, the pufferfish, that's venomous, and that venom, it's called tetrodotoxin, is a molecule that's shaped just so that it fits right here in that voltage-gated sodium channel pore so that sodium cannot enter, and so it blocks the conduction of action potentials. Cocaine works the same way, but not to produce its stimulant effects. Cocaine has other effects on the nervous system, including a local anesthetic effect, so it has a numbing effect. And cocaine works the same way. It drops right here in the pore of the voltage-gated sodium channel. Lidocaine, which is the generic name for Novocaine, which is a trade name for the drug that dentists use to numb, parts of your mouth when doing dental work, works exactly the same way. It sits here in the pore, blocks sodium from entering, and so prevents the conduction of action potentials. And if those are carrying pain information, right, or pressure information, then it is not going to get through. It's not going to be encoded, and you won't feel anything. We'll talk about the other mechanisms of cocaine at a later time. Drugs can affect the storage of neurotransmitters. There are proteins that are involved in taking up transmitter molecules that are here in the cytoplasm and packaging them into vesicles. This is one way that amphetamines work on the neurotransmitter dopamine. There are drugs that can interfere with the release of neurotransmitter by interacting with the voltage-gated calcium channels that are necessary to dock the vesicles to the presynaptic neuron. There are presynaptic receptors. We've talked about the postsynaptic receptors for neurotransmitters, but there are presynaptic receptors that are expressed, and we'll talk about those later on in the course, and drugs can interact with those as well, and this can change the release of neurotransmitters from the presynaptic neuron. Most of you are familiar with the process of neurotransmitter reuptake, and there are proteins that are expressed on the presynaptic neuron that grab transmitter molecule inside of the synaptic gap and draw it back up. They take it back up into the neuron that released the transmitter. This is one of a few ways of shutting down the signaling of transmitter release. So once you send that signal, you want to stop it so you can send another one in rapid succession. So one way to stop that signal is just to remove transmitter molecule from the synaptic gap, and these proteins do that. They're called transporter proteins, and you've probably heard about transporter proteins that are selective for specific transmitter molecules, and drugs that interact with those proteins, like the selective serotonin reuptake inhibitors. Prozac, Zoloft, and Paxil are trademark names for those drugs that block the transporter that's selective for serotonin in the synapse. That means that more serotonin is going to stay in the synapse for a longer period of time and interact with postsynaptic receptors for a longer period of time. Another way of stopping the signal of synaptic transmission is that enzymes are continually breaking down transmitter molecule in the synaptic gap. And there are drugs that interfere with or enhance that metabolism. The earliest antidepressant drugs, we talked about Prozac, Zoloft, and Paxil, but drugs that appeared before those, in fact the first antidepressant drug blocked the metabolism of lots of different types of neurotransmitters like serotonin, dopamine, and norepinephrine by interfering with an enzyme, right, a protein that's called monoamine oxidase. So those monoamine oxidase inhibitors were drugs that slowed down the metabolism of transmitters like serotonin and dopamine, norepinephrine, and kept those transmitters in the synapse for a longer period of time. So there's many, many presynaptic mechanisms of drug action. We can talk about postsynaptic mechanisms of drug action as well. So this is the receiving neuron now that's expressing different types of receptors for neurotransmitters. And one of the simplest postsynaptic mechanisms of drug action is that there are drugs that mimic neurotransmitters. They're shaped very much like them. LSD and nicotine are examples of these. LSD fits into a space on a specific type of serotonin receptor and activates it as though serotonin were there. Nicotine looks very much like acetylcholine to a specific type of acetylcholine receptor, and it fits there and operates the receptor just as though acetylcholine were there in the receptor. So when a drug mimics that neurotransmitter and operates the receptor normally, we call that an agonist effect. So we can say that nicotine is an acetylcholine receptor agonist. We can say that LSD acts as a specific type of serotonin receptor agonist. Similarly, you can have a drug that fits into the binding site for a neurotransmitter but does not operate the receptor. normally. In fact, it blocks the action of the transmitter. So if we have a dopamine receptor, there are several drugs that act to block the binding site for dopamine, but do not operate the receptor normally. We call this an antagonist effect. So we would say that drugs like, well, there's certain neuroleptic or antipsychotic drugs like Thorazine, which is a trade name of a drug called chlorpromazine, that they block dopamine, a couple of different types of dopamine receptors, so that when dopamine comes along, it cannot fit into its binding site. We'd say that that's a dopamine receptor antagonist or blocker. There are other types of receptors that don't pass ions, but rather send a signal within the postsynaptic cell, an intracellular signal. There are drugs that interfere with the signaling inside the cell, which we'll talk about in more detail later on, but lithium is an example of one of these drugs that, that modulates the effect of these messengers we call second messengers that send signals inside of cells in response to transmitter being bound on the outside of the cell. Oh, one question that we've had in class is, well, where do these drugs come from? Are they packaged into vesicles and released into the synaptic gap? And the answer to that is no. The drugs come from the blood supply, and they go all over the body to all the organ systems, including the brain. And they diffuse into the extracellular space and get into the synaptic gap that way, where they can interact with receptors and other mechanisms, both pre and postsynaptically. One last thing. In the continued presence of a drug, this postsynaptic cell doesn't just sit there. It can respond by changing the number of postsynaptic receptors, and this would be a chronic effect of drug treatment. In fact, we're pretty sure that that's how some of the antidepressant drugs that we talked about work. So those antidepressants that act on presynaptic transporter proteins can have postsynaptic effects over the long term. Another example of this is in smokers. So people who are continually flooding this space with nicotine, this postsynaptic cell produces receptors that are less sensitive to acetylcholine and thus to nicotine as well. So you can change the number of postsynaptic receptors, and we'll talk about that as a mechanism of tolerance for drugs in the future. All right, that's it. We'll see you in the next video.